matrix assisted laser desorption ionization time of flight mass spectrometry or MALDI-TOF-MS. MALDI-TOF-MS has been applied for the analysis of a wide range of biomolecules. The term MALDI was coined in 1985 by Franz Hillenkamp, Michael Carras and co-workers. They found that the amino acid alanine could be ionized more easily if it was mixed with the amino acid tryptophan and irradiated with the pulsed 266 nanometer laser. The tryptophan was absorbing the laser energy and helping to ionize the non-absorbing alanine. Peptides up to the 2843 Dalton peptide melittin could be ionized when mixed with this kind of matrix. In MALDI, the sample is incorporated into matrix molecules and then subjected to irradiation by the laser. The laser promotes the formation of molecular ions. The matrix is typically a small energy absorbing molecule such as 2,5-dihydroxybenzoic acid or alpha cyano 4-hydroxycinnamic acid. The analyte is spotted along with the matrix on a metal plate and allowed to evaporate, resulting in the formation of crystals. The plate is then placed in the mass spectrometer and the laser is automatically targeted to a specific places on the plate. Since sample application can be performed by a robot, the entire process including data collection and analysis can be automated. This is the single biggest advantage of MALDI. Mass analysis follows the conversion of proteins or peptides to molecular ions. This is accomplished by the mass analyzers in the mass spectrometer, which resolves the molecular ions on the basis of their mass and charge in a vacuum. In MALDI-TOF, the ions termed in an ion source are accelerated in an electric field and thereafter propagate through the field free region before reaching the detector. When entering the field free region, all ions of the same charge state have obtained approximately the same kinetic energy, which is defined by the acceleration voltage. Hence, low mass ions will travel faster and hit the detector before heavier ions. A TOF spectrum is obtained by measuring the time elapsed from the laser pulse to the detection of the various ions having propagated through the field free region and by measuring the intensity of the detector signal at each time. Summation of TOF spectrum from several laser shots is typically performed. A mass spectrum can be obtained by converting the TOF to mass by Z using the fundamental relations between these quantities via a calibration procedure. To improve resolution, MALDI-TOF instruments are often equipped with a delayed extraction facility. Reflector MALDI-TOF instruments are equipped with an electrostatic mirror to improve the resolution. MALDI-TOF-MS has the advantage of showing all the under-methylated products which are insufficiently volatile to be observed by GC-MS analysis. MALDI techniques typically employ the use of UV lasers such as nitrogen lasers 337 nanometer and frequency tripled and quadrupled ND-YAG lasers 355 and 266 nanometer respectively. Although not as common, infrared lasers are used due to their softer mode of ionization. The laser is fired at the matrix crystals in the dry droplet spot. The matrix absorbs the laser energy and it is thought that primarily the matrix is dissolved and ionized by addition of a proton by this event. The matrix is then thought to transfer proton to the analyte molecules, example protein molecules, thus charging the analyte. Ions observed after this process consist of neutral molecule and an added or removed ion. Together they form a quasi-molecular ion, for example, M plus H plus in the case of an added proton, M plus Na plus in the case of an added sodium ion, M minus H minus in the case of a removed proton. MALDI is capable of creating singly charged ions, but multiply the charged ions M plus NH and N plus can also be created as a function of the matrix, the laser intensity and or the voltage used. One of the challenges in the development of the therapeutic antibodies is the development of a fast and precise method to determine the molecular weight of antibodies. Historically, gel analysis, size exclusion, analytical ultracentrifugation and multi-tryptophan have been used to determine the molecular weight of their entire molecule or if its separate components. However, each of these methods suffers from either imprecise molecular weight determination or from the lengthy time required to perform the analysis. Mass spectrometry based approaches for targeted protein quantification are based on the concept of isotope dilution mass spectrometry techniques commonly used for the detection of small molecules. The approach is comparable to modern advanced antibody based methods such as the multi-analyte profiling platform MAP, offering multiplexing detection capability with an excellent dynamic range. While the antibody development is central to the success of an ELISA based platform, MS provides alternative and complementary approach which can precisely detect a wide variety of peptides or proteins via high mass accuracy and or peptide sequencing. This approach provides high selectivity and specificity and technically avoids most of the problems associated with optimization of multiple assays in a single measurement. The mass spectrometry based technique has a unique capability to perform candidate based proteome browsing and measure absolute levels of post-translational modifications, demonstrate the general components of mass spectrometry based approaches for targeted protein quantification. An antibody is a large Y-shaped protein used by the immune system to identify and neutralize foreign objects such as bacteria and viruses. Each antibody recognizes a unique antigen target. It consists of two identical light chains and two identical heavy chains held together by interchain disulfides and non-covalent interactions. Furthermore, post-translational modifications occur during cellular development and production, and these can affect the properties of the antibody. For example, if the amino terminus of the heavy chain starts with a glutamine residue, it can be converted to pyroglutamate. Also, on the heavy chains, the C-terminal residue is often a lysine residue that may be post-translationally excised. Of additional importance is the addition of various glycans to an asparagine residue in the heavy chain, generating an N-glycan linkage. mAbs and their modifications need to be characterized quickly and accurately as part of bioprocess development for the evaluation of product consistency. A critical component of characterization is an accurate determination of the antibody's molecular weight, which is important because the mass provides a glimpse of the integrity of the structure. Future prospects. The prospects of this technology are exciting and promising. It is a long journey to develop a robust, high-throughput multifunctional system that can provide sophisticated function that we desperately need in the clinical diagnosis and therapy. Most of the current spectrometry-based platform technologies require a combination of multiple sample preparations such as immuno-subtraction, multidimensional LC separation, immunoaffinity and or solid phase extraction to enhance the analytical dynamic range and detection sensitivity. These requirements limit the capability for high throughput. Thus, the major caveat for the current mass spectrometry based approaches for targeted analysis is the lack of a high throughput pipeline integrating a highly specific and ideally single step sample preparation strategy. The development of a quantitative detection technology that provides high specificity and sensitivity, multiplexing, absolute quantification and PTM monitoring should provide us a powerful tool for a broad range of applications and greatly enhance our effort in developing better medicine.